Hey, welcome to my office. Since you guys haven't been coming by to visit me here, I just wanted to talk about proteins. I felt like I didn't finish saying what I wanted to say at the end of the lecture. By the end of the lecture, and we're moving on, of course, since we're always doing another chapter in this class. So, as we talked about, proteins are an example of a polymer. So the monomer, in this case, what we're stringing together, lots and lots of, the monomer is an amino acid. There are 20 different amino acids. We're sticking them together in different orders. So that's the brief introduction for what I want to talk about. Let's go ahead and jump right into it with some thoughts on proteins. Okay, so when you think of proteins, a lot of times people just think of things like steak and chicken and protein as something to eat, and that's true. We like to eat muscles from cows and chickens and other animals, and yes, they have some nutrition value for us. So they have proteins, and we can take apart those proteins, break them down in our digestive system into individual amino acids, and then put those amino acids into our bloodstream, deliver them to our cells. We can use these amino acids to build proteins of our own. Now, granted, of the 20 different amino acids, we can make all but 8 of them by ourselves, just out of carbohydrates and things. We can just modify molecules and build our own amino acids. But there are 8 of them that we call essential, meaning that it's essential that we get them in our diet, and because we can't make them. It's just a vertebrate thing. It's not just us, it's not just humans. It's pretty much the whole vertebrate, fish, amphibians, reptiles. We just have problems with those 8. That's okay. You can eat meat if you want, and that's a complete protein. It has all the different amino acids in it, or you can just mix things up with vegetables and make sure that you get a complete assortment of proteins. I probably shouldn't have taken off my shirt for this one, but so the muscles in our cells, in our bodies, are pretty similar to the muscles in chickens and cows and things. It's just there. That's a little better. Boy, that guy's head looks too small. Anyway, muscle has lots of protein because that's how muscles move. It's actin filaments and myosin, and it comes down to the cellular level. Every time you twitch a muscle, there's a lot of activity going on between proteins. So the proteins are responsible for moving all of your muscles. But it goes beyond that. Your tendons are made out of collagen primarily, and collagen is a type of protein. You can see some collagen molecules there that are, kind of a rope-like sort of protein. Keratin, skipped over my keratin. Hair, fingernails, horns are an example of keratin, and this is just ribbons and ribbons of keratin molecules, these large proteins that are structural in nature. Keratin is pretty interesting because it's quite strong. Don't think of hair as being that strong, but you think of horn as being strong, and nails maybe are intermediate. But they're all made with large quantities of keratin, and keratin has a lot of cysteine, and cysteine has sulfur, and different cysteines can be bound together with what's called a disulfide bond. And these disulfide bonds are responsible for the strength of the keratin. So human hair is about 17% cysteine. So that's just one amino acid. So you would think that if it was just average, you'd have 1/20th about of a protein would be cysteine if it was just kind of random. But no, you have almost 20% of the protein is made of cysteine. So lots and lots of disulfide bonds in this particular protein. And by the way, when they do things to hair, burn it, or if you go to a hair salon and you smell, like if they do a perm, it stinks because of the sulfur. They're trying to make more disulfide bonds when they curl the hair, they want it to stay in place there. They're trying to add more disulfide bonds to the actual keratin molecules. So anyway, that's smelly because of the sulfur, or when you burn hair or horns, it would be smelly for that reason. Blood doesn't do what it does except for the hemoglobin. and hemoglobin is an example of a protein. It's a protein, it's 4 proteins put together to form one hemoglobin molecule, and each red blood cell has tens of thousands of hemoglobin molecules, each one carrying 4 oxygen molecules. So it's just another example of what proteins do. And they do more than that. They don't just move muscles and provide structure and carry oxygen. Perhaps the most important role is that they act as enzymes. Okay, and let me move my head down here. So with enzymes, you know that an enzyme is a biological catalyst. A catalyst is something that speeds up a chemical reaction by lowering the activation energy. If that doesn't make sense, then come talk to me about this, because it's pretty important. Enzymes don't just speed up reactions a little bit, they make them go 1,000 times faster, or 10,000 times faster, or 1,000,000 times faster, or sometimes 10 million times faster. So reactions that would never take place at the speed that life needs them to take place at, they happen all the time thanks to the different enzymes. So enzymes, they have an active site where the substrate fits in, and it's very specific. And this is just a kind of the lock and key model, where here comes the substrate, it bonds to the enzyme in a temporary fashion, and there's some stress put on the substrate on particular bonds so that it breaks into 2 products, or it could go the other way. These 2 products, these 2 molecules could be the substrate, and you could then join them together, right? It goes both directions. Enzymes are not used up in chemical reactions. They get used over and over and over, and it can go one direction or it can go the other direction depending on the conditions. So what else do we say? I mean, enzymes are just incredibly important. It's the shape really matters. You know, they've shown it here that there's this perfect match between a substrate and an enzyme, and here you can see that there's the active site, and that the enzyme may actually change shape a little bit. It conforms, and when the 2 come together, the substrate may cause the enzyme to change shape and act on it. Anyhow, the shape really matters with proteins, and if you change the shape, then the protein's not going to function anymore. So you can denature proteins by changing temperature too much, or especially increasing the temperature, or by changing the pH. And this is one way to die. Your blood becomes too acidic, or you get too hot. You get stuck outside in the summertime and you overheat, and your enzymes may cease to function. Okay, so the shapes of proteins are really complex, and we're starting to understand them better, but we have a lot to learn about shapes of proteins, and how these shapes matter, and how they actually work at the molecular level. So in talking about protein shape, we start by just talking about the primary structure, which is just the order of the amino acids. Now this one's easy because we can just look at the genetic code. Every 3 letters is a codon, which codes for one of the 20 different amino acids, and we just simply we build the protein. We start, it goes from, there's a directionality to it. So there's an amino end and a, can't remember what the other end's called. Anyhow, there's an order, and you can just write whatever random genetic code you want, send it to a lab, and you can have your own custom protein if you want. It won't necessarily do anything. It almost certainly won't do anything, but you can make your own proteins, custom proteins. So the idea is, well, let's make a protein that actually can do something, right? So maybe I find a gene for a particular cellular enzyme, and I say, hey, I want to put that enzyme to work. I'm going to build this protein. I'm going to have my little tool. But just having the right sequence doesn't mean it's going to get folded into the right shape. That's the problem. The primary structure is easy to get. We can get the primary structure correct, but that doesn't necessarily mean that it's going to fold properly. It may get some of the secondary structure on its own. So you get these pleated sheets and these alpha helices. And by the way, I didn't put a slide in about this, but these alpha helices are non-polar. And so when we look at embedded proteins that are embedded into the plasma membrane, so remember how a plasma membrane is a phospholipid bilayer, So here are phosphate heads and here are phosphate heads, and in between there are lipid tails. And sometimes proteins, get embedded into the bilayer, and so you'll have a part of the protein that's sticking up above and maybe a part that's sticking into the cell. And we have this section in between where the protein fits in there. It's usually an alpha helix because the alpha helix is nonpolar, so it fits in nicely to the nonpolar lipid tails. There's also what's called a beta barrel, which is a bunch of beta pleated sheets in kind of this weird spiral fashion, and it's a bigger structure. There's a beta barrel is another way to get into the membrane, but it's usually alpha helices, and a protein may have just one alpha helix going through the membrane. It may have multiple alpha helices. The protein may have several different loops, and it might cross through the membrane in several places. So in the lab, we can get a lot of the secondary structure, so that's not the hard part. The hard part is getting the final three-dimensional shape, getting the disulfide bonds in the right place, just getting things to bend and fit together in the proper shape. That's the hard part. So there's a tertiary structure that we just can't get too well in the lab. And the quaternary structure is if we're joining two or more proteins together. Now there can be some processing that needs to happen in the Golgi apparatus, some slight modifications that need to happen to activate the protein to get it into the right shape. But within the cell there are tools for making these tools. Tools that help the proteins fold into their proper shapes. So it can be impossible at this point in the lab sometimes. So what do? Well, for human insulin as an example, so we use human insulin to help fight diabetes. So people who are they're insulin dependent diabetics. Used to be that we'd have to try to extract insulin from horse serum, and that was very expensive, and you didn't get much insulin and had all sorts of problems. We were able to take the gene for human insulin and stick it into a plasmid, and then stick that into a bacteria, and then we have this recombinant DNA, this bacteria that has human gene in it, and it's the gene for making insulin. The bacteria has no use for insulin, but you get the gene in there and it starts producing insulin. And basically you can just then feed these bacteria sugar and nutrients and grow them in these big fermentation chambers, and they'll just crank out the insulin, and then you can just harvest it, which is great. So I don't like my head in either place there. Stick my this is just another example. When your blood clots, there are a whole bunch of different clotting factors that get involved. It's a clotting cascade. One protein activates another, which activates another, and blah 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 blah. And boom! In the end, you get this fibrin clot, this protein that forms. It was in solution, it comes out of solution, and it clots your blood. People who are hemophiliacs, who have bleeding disorders, it's often a problem with clotting factor 7. It's just not the right shape, so it doesn't work properly. So they tried sticking this into bacteria. The bacteria starts making the protein correctly, in the sense that it puts the correct amino acids in the correct order. Maybe the secondary shape is correct, but the tertiary, the final three-dimensional shape, doesn't work properly. So it didn't work. So it worked for insulin. It did not work for clotting factor 7. We don't know, what went wrong exactly, probably, but the bacteria just didn't quite have the right machinery. So we tried it in yeast, because that's a eukaryotic cell, and still didn't really work. So what do? In the end, what worked was $1 million goat, right? This goat, a little pygmy goat here, we get the gene inserted into a region, that codes for milk. So I think it's, for when the goat expresses milk, there's some milk sugars. I think it's right in that region of its gene. But old MacDonald had a farm, play on words there. So basically one goat can crank out enough clotting factor 7 to save, I don't know how many people, a lot of people. and it works very well. So I just wanted to say a little word about the protein shape and how we actually study it, because sometimes it's important to know the shape, because then you can maybe make a drug that will fit a particular protein. Maybe you want to turn off a protein or mimic a protein or do something, and you need to know the shape. These are just 3 different ways to view the same shape. This is a ribbon model or a backbone model or a space filling model. There are lots of different ways that you can view the shape. I don't know if one's necessarily better than another, but I wanted to tell you a little story. When I was in high school, I visited a lab that was studying the shape of a particular protein. It was an enzyme, I don't remember which one, but I was handed a little tiny vial with a little crystal inside of it. It looked like some salt inside. And I was like, huh, that's interesting. It really wasn't interesting. And I was gonna, kind of shake it around, and the guy's like, oh, careful, it took us 600 tries to get that protein to crystallize. And, we've worked on it for 6 years or something. And I'm like, well, why did you hand it to a kid? So anyway, I gave it back to him, and what were they doing with it? They were shooting x-rays at it, and they had some setup that looked kind of like this, and from that they get data that looks like this. That as the x-rays hit the different amino acids, they scatter, they give some sort of reflectance pattern. This means nothing to me, but they then interpret that, and they would make 3-dimensional models trying to get the protein. And I couldn't find a picture of what I saw exactly here. This is a pretty big model that they've made here, but this was in the late 80s that I saw it. They had a whole room, a big room, like as big as a classroom, that had the model, and they had styrofoam balls of different sizes for the different atoms. So carbon was this big, and hydrogen was this big, and oxygen, and they had them painted different colors. They had every single atom of the protein mapped out in 3 dimensions, and it took up this whole room. And I was like, wow, this is cool. And they're like, oh, you want to see really cool? And they flipped out the lights, and they turned on the UV lights, and now all you could see were these glowing balls floating in 3 dimensions, and we could walk around, and they're like, ooh, see this? This is the active site region. And we think that over here is where the coenzyme fits in. And they had spent years and years and years constructing this model, and it was really impressive. So nowadays they have much fancier machines, and don't ask me to explain this, but it looks something like this, and they shoot x-rays at it from all different directions. The data are similar, but now they have computers to help understand and interpret it, and understand the electron density, and interpret the final 3-dimensional structure. So they started the protein bank, I don't remember which year, not too long ago. They had 7 structures. Now they have over 100,000 proteins logged in. And so I would encourage you to go to rcsb.org and just play around. And one of the easiest ways to play around is to go to the search, and then under search there's a browse by annotation, and you can just pick membrane proteins, protein modification, protein symmetry, biological process, enzyme classification, and you can just choose any of these. And then if you click on any of the blue, anything you see in blue, it will pull up that particular protein, and then you'll have all sorts of cool options of what you can do with it, different ways to view it. So these are different. I was playing around with it, and you can turn them around in 3 dimensions, and you can you can right click on the protein and get all sorts of options, and just play around with it at rcsb.org. Well, that's pretty much it for today for talking about proteins. Hopefully that was a little bit helpful. Take-home message: proteins are the tools for running the chemical laboratories that are our cells. So in chapter 3 we're going to talk about cell metabolism, which is all the chemical processes happening within a cell. Just remember that for every process you need lots of tools, and those tools are enzymes. Those enzymes are proteins. So you have, and it's confusing because we only have about 25,000 genes, 23,000 genes. Okay, but we're able to make over 100,000, 200,000 proteins, something like that. We have incredible protein diversity. So that's pretty confusing, because for a long time we thought one gene codes for one protein, end of story. How could you have more proteins than genes? So that's just, I'll just leave that as something for you to think about. Okay, all for now. I'll talk to you later.